Problem. Hi, everybody. I'm Farida Akhtari. Can you guys hear me? Okay. I'm a statistical geneticist at the National Institute of Environmental Health Sciences, which is a part of the National Institutes of Health here in the US. And I'm going to give an overview of the PEGS Dream Challenge um, before our winners come and talk about their exciting models that they submitted, their analysis and results. So the goal of the PEG Stream Challenge was to find the genetic factors, the environmental factors, and their interplay that affect human disease. Specifically for the challenge, it was high cholesterol. And we were really excited to use the Dream Challenges platform and partner with Sage Bionetworks to host the PEG Stream Challenge because Dream has conducted several such data challenges focusing on a broad set of healthcare and life sciences questions, which have attracted a large multidisciplinary international community and has also led to high impact publications. So as I mentioned earlier, the goal of the PEG Stream Challenge was to understand the etiology of high cholesterol using the multidimensional genotype, phenotype, and environmental data from the personalized environment and gene study, that's PEGS, to improve our understanding of high cholesterol. And PEGS is uniquely poised because we have environment, diet, lifestyle, genomics, and geospatial data for a diverse group of participants in the US. The challenge was launched on May 1st, and it concluded very recently, August 31st, and we announced the challenge winners on September 1st. So other than bragging rights of winning the Dream Challenge itself, of course, all the challenge participants will be included as authors in the manuscript, which we will write very shortly, describing the PEGS Dream Challenge. And one of our winning team is here to present at this conference, and another one will be presenting remotely as well. So I think that's really exciting. And for anyone who needs more information on the PEG Stream Challenge post this presentation, that's the challenge wiki for you to look into further. So for the PEG's Dream Challenge, we had two separate distinct tasks, and participants were, they could participate in one or both tasks. The first task was disease classification. So the goal of this task was to classify high cholesterol status in the held out test data set for the PEGS participants using any combination of health, exposomics, geospatial, and genomic data from the PEGS data set. And the submission included Docker container models and a write-up as well. And the evaluation criteria was AUC to assess model accuracy. The second challenge was an ideation challenge. This is a really interesting challenge. The goal of this challenge was to harness the collective creativity of the challenge participants to generate really fresh and novel ideas, to generate new hypotheses in order to further understand high cholesterol etiology. And so challenge participants were asked to develop a novel hypothesis or generate a model. And the focus for this challenge was to really use the multi-dimensional aspect of the PEGS data. So genetics, environmental data, geospatial data. We really encourage participants to focus on the multidimensional aspect of the PEGS data for this particular task. And submissions with working models or at least detailed or specific mathematical model descriptions were highly encouraged. The submission for this task was a write-up, and these were evaluated by a panel of judges based on scoring criteria that consisted of creativity, significance, how interpretable the model was, how innovative it was, and potential translational impact. I'll describe the PEGS challenge data in detail in the next few slides. So this is the data from the PEGS study, and all of the data was available as part of the PEGS Dream Challenge. So as I mentioned, PEGS is a multi-dimensional data set. It consists of genotype, phenotype, and environmental data. I'll explain some of these data components in the next few slides. PEGS is a long-term health, exposure, genomics, and geospatial database, making it a really great resource for investigating the interplay of genetics and environment and how they influence disease status. And it's a really mature data cohort with analysis ready data, and we worked really hard over the last 4 years in establishing really good data sharing practices and also including comprehensive documentation with the data. These are the PEGS participants' demographics. You'll notice that we have a lot of diversity across age, sex, race, ethnicity, and socioeconomic status in PEGS. Most of the participants are from North Carolina. We have almost 28% Black individuals and roughly 33%, roughly 63% female participants as well. And participants are actively engaged in the study, and they are available for callbacks and add-on studies if required. There are 3 main surveys in PEGS. The first one is the Health and Exposure Survey. This survey consists of self-reported disease status. You see a snapshot of some of the diseases here, like some of the highly prevalent diseases in PEGS, from diabetes to asthma, high cholesterol, blood pressure, allergies, etc. And the prevalence of these diseases in PEGS is very similar to the prevalence in the US. So the PEGS cohort is quite representative of the US population. In addition to the disease status, the Health and Exposure Survey consists of family health history, socioeconomic status, lifestyle data, and occupational exposures. The next 2 surveys are focused on the exposomes. So here participants are asked specific questions about what kind of exposures they have been exposed to. There are 2 surveys. The first one is the internal exposome survey, which consists of exposures that are internal to your body, so like medication, sleep, your diet, exercise, etc. And the second survey is the external exposome survey. So what kind of chemical exposures, occupational exposures, workplace hazards, are you close to certain hazardous sites like nuclear power plants or gas stations? What kind of pets do you have? Do you live close to some agricultural or farm sites? Are you exposed to some occupational carcinogens, etc.? So this is a really rich data set of exposure questions. Each of these surveys is roughly 700 questions each. The next data type in PEGS is geospatial data. So we have participant addresses from 5 different study events. And each of these addresses has been geocoded to latitude, longitude, and then these geocoded participant addresses are linked to various geospatial databases. So hazardous sites, MERA2 data, the Social Vulnerability Index, and the Environmental Justice Index. So we have all of these GIS linkages available, and these are also substitutes for environmental exposures that can be used for analysis. We also have a really rich genomic data component in PEGS. So a subset of the PEGS participants, roughly 4,700 participants, have been whole genome sequenced. So for these participants, we have called single nucleotide variants and structural variants and HLA genotypes as well. We also have estimated telomeric content, and we also have local and global ancestry estimations for these participants from the whole genome sequence data. The same participants have also been sequenced using the EPIC chip, so we have methylation data on 4,700 participants as well. And all of these genomic data components were also available for the data challenge. So to summarize all of the PEGS data that I just showed you in graphic format, we have in the first column, we have all of the surveys. So this is self-reported data of health and exposure and exposomic questions. Then we have the genomic data set in the second column, consisting of all the various calls that we made from the whole genome sequencing and the methylation data. We have geospatial data, and then the phenotype for the PEGS Dream Challenge was high cholesterol. Note that the PEGS cohort and the PEGS data set itself is not of just one phenotype. We have many different phenotypes that have been reported by the participants. But specifically for the data challenge, we wanted to focus on one phenotype. Next, I'll talk briefly about the challenge details. So as you can see, all of the challenge data comes from PEGS participants, and so this is PII. So we were very conscious of protecting the PEGS participants' privacy. And so for that, we de-identified all of the data. All of the PII and PHI data was removed from the challenge data. And we also anonymized the data. So all participant IDs were replaced by fake IDs in all of the data components that I just showed you in the previous slides. Further, the original PEGS data was not available for download for the challenge. But what we did is we created a synthetic data set, which was very representative of the original data, and this data was available for download. And this synthetic data was provided for model construction and code development purposes. But the submitted models that challenge participants submitted into the SAGE infrastructure, those were run on the actual PEGS data. So all of the actual real PEGS data was on the SAGE servers, and submitted models or Docker containers ran on the actual PEGS data. And so challenge participants received feedback on their model performance on the real PEGS data. All of the data was QC'd, filtered, and analysis-ready data so that challenge participants didn't have to waste time doing QC or filtering. Our goal was to empower the challenge participants to just focus on model creation, coming up with interesting hypotheses, and interpreting their results. So we did a lot of work prior to the challenge to ensure that the data was analysis-ready. We split all of the data, or each of the actual data and the synthetic data, into training, validation, and test sets, and conducted extensive QC of both the challenge data sets before challenge launch. We also did a proof of concept run prior to the challenge start to ensure that there was enough signal in the data for the phenotype of interest, which was high cholesterol. So our analyses, we did a GWAS, XWAS, and EWAS, or methylation analysis, all of which showed high signal for high cholesterol phenotype. And we also did a POC, or a test run, on the SAGE infrastructure to ensure that all of the data can be successfully run on the SAGE infrastructure, because some of the data components are pretty large. We created a staging site with the challenge data, with all of the information, and we did all of our test runs on the staging site. We created evaluation criteria for each of the challenge tasks that I briefly mentioned earlier. And additionally, we set an AUC threshold for classification accuracy for the first challenge task. The idea behind this was that we wanted to encourage participants to explore novel and innovative approaches to integrate the multidimensional data components in PEGS. And we really wanted them to use the health, exposure, genomics, and geospatial data to surpass a classification accuracy, which was 0.73. And this AUC was obtained from a model using a polygenic score as the main predictor for high cholesterol. And this polygenic score was computed for the PEGS participants using the metadata from the high cholesterol polygenic score from the polygenic score catalog. This figure shows the challenge workflow. So we have all of the PEGS data, the actual data on the left-hand side, the phenotype, genotype, and environmental data for roughly 9,000 participants. We created a synthetic copy of the PEGS data, and each of these data sets were divided into training, validation, and test. During the leaderboard phase, the challenge participants were able to submit 5 submissions per day in order to build their models and develop their workflows and get model performance feedback. And this phase, after this phase was completed, we were in the final round. And during this round, challenge participants could submit their final models, which were run on the held out test data set. And this is where they were scored for challenge task 1, and an AUC was computed for each of the submitted models. So this was all of the challenge data. We also created a lot of challenge resources for the challenge participants that they could use. So we had the PEGS Dream Challenge Wiki with a lot of detailed information about PEGS, the PEGS data, links to various resources, challenge rules, submission guidelines, and evaluation criteria. I created a FAQ page to address some of the idiosyncrasies of the PEGS data, because there are some some things that are very specific to the PEGS data, and some questions that I know have come up from our collaborators that we worked on, that have worked with us with the PEGS data. So we wanted to make it easy for the challenge participants to access those kind of questions easily on the FAQ page. We had a discussion forum as well, and we were happy to receive any questions and help out challenge participants with whenever they were stuck with something. We also provided the challenge participants with various resources for PEGS for data exploration, code model development, data extraction, data visualization, data formatting, etc. So a couple of them are listed here. So we have the PEGS utility library, which contains like common utilitarian functions for ingesting the PEGS data, formatting it, harmonizing the data across the different data components, etc. We also have a bunch of example scripts for working with the geospatial data. So we made those publicly available for the challenge participants. We didn't want them to reinvent the wheel every time, so we just made it easy for them to use the data for their analysis. We also have public access to the PEGS website that you see on the right. That's a public website by NIEHS that anyone can access, and I encourage you to visit the website if you want more information about the PEGS study. Additionally, we also have the I2B2 web client for data exploration. We also have the PEGS XWAS results. These contain results of our published XWAS study that we've done in PEGS for about 15 different phenotypes of the PEGS participants. And this is publicly available. If you go to the PEGS website, you can look up the graphical representation of the PEGS XWAS. And we also provide like correlation globes that allow you to look at the correlations between exposures and disease, and they are visualized using those lovely correlation globe figures. I'll briefly talk about the challenge results. I know our challenge winners are here to go into detail about their model submissions. For task 1, we had several exciting submissions. So again, the task 1 was to classify high cholesterol status in the held out test data set using any combination of the PEGS data components. So participants were free to use health, exposomics, geospatial, genomic data, whichever component or combination that they wanted to use. So we had several submissions. Submissions included adjusted neural network models that consisted of a main model that used the exposome surveys and auxiliary models that added the health and exposure information to the main model. We also had models using XGBoost, gradient boosting, classification trees. We had models identifying feature importance. We had multi-layer models using random forest, on the exposome surveys and combining results from that model with polygenic scores. So I thought that was really interesting. The results from the various submitted models showed that the data from the self-reported surveys, the health and exposure, internal and external exposome, provided really informative predictors for high cholesterol status, in addition to the well-known clinical and genetic risk factors for high cholesterol. Task 2 was the ideation challenge. This was the open-ended challenge to generate novel hypotheses and gain new insights about high cholesterol. And again, here we had some exciting models that used G by E. We had staged analysis models utilizing survey data, whole genome sequence data, and HLA genotype data. So combining the different data components as was intended for this challenge. And we had models that use multimodal neural networks as well. So I think we had some really good submissions for this challenge as well. So in conclusion, we had a really exciting and successful data challenge with the PEG study. We had 90 challenge participants from across the globe, across 5 different continents. We covered all continents. I think we missed out on South America, but maybe next time. We had 345 submissions across all rounds of the challenge with several interesting ideas and methods that were submitted. And we will be collating all of this work into a manuscript describing the challenge. And we're really excited to see that the challenge itself fostered collaborations amongst participants who joined from various different institutes to form a team for the challenge. And we hope to receive collaborations for the PEG study even after the conclusion of the challenge. And the PEGS study highly encourages collaborations in the research and scientific community. So if you are interested in the PEG study and its data, please feel free to request access to the PEGS data. We have a very streamlined and short process. All you have to do is fill one form at the link that I've listed up here, and you sign a DUA with NIEHS, and we give you access to the data. So I would like to thank, first and foremost, the PEG study participants who provided their information for the PEG study, and financial support was received from the intramural funds from the NIEHS. We want to thank the SAGE BioNetworks and the DREAM Challenges organizers, and Gaia, Verena, and Jake, who helped us with the SAGE infrastructure and in organizing the DREAM Challenge, and the ICSP conference organizers as well. And we want to thank all the participants who joined the PEGS DREAM Challenge and made it a success. With that, I'm happy to take questions.
Download, they had access to synthetic data. They didn't download the actual data, right? So then the models, so then they submitted a pipeline to train models and generate the models. So, but they really didn't really see the training data. Is this?
That's only partly true. So they were only able to download synthetic data, which was essentially like a fake data, but it was representative of the actual data, meaning it had all the data components, all the fields. Right. Its structure was exactly the same as the real data. But when they submitted a model into the SAGE infrastructure, like when they submitted a Docker container, that was run on the actual PEGS data, on the real training data. Right. And they got feedback. So they got a feedback saying, okay, your model ran, its AUC is 0.6 or something. And they could then improve their model based on that.
So they did not submit models, they actually submit a pipeline that generates models.
Yes.
All right. So that's, it's even more stringent than what we did, where we just, the training, they could train on it, but they had to submit the model. You are saying, no, they submit the script that generates the model that will be tested.
Yeah, they submit a script that builds a model that.
And one more quick question. So would you have also information about if any of those women were pregnant, and would you have that kind of information?
Yeah, we do actually. We have pregnancy information in one of the exposome surveys in multiple surveys, we have information on pregnancy and then complications due to pregnancy, etc. In fact, one of the studies, if you go to the PEGS homepage, one of the papers that has been submitted from our group, a really talented PhD student in our group has worked on hypertension complications in pregnancy,
Right.
and she's found some really interesting results that have been published. So I think one of the messages from the PEG study is that even though we are smaller than some of the bigger biobanks, the data is really rich and has a lot of signal. And even with such a small number, not such a small, but a smaller number of participants, we found really interesting results.
Thank you. Very kind.
Thank you. Really incredible data set. Adi, we did run previously challenges for which you could train in private data. For example, a digital mammography challenge. Unfortunately, everybody used some other data they have private, and they didn't really train with that. I mean, maybe they're refined, but we have in the past. But this was. But you mentioned that people could also visualize and explore the actual data.
Yes. So there were several ways to do this. Here, so we have the PEGS website. All of this, again, is public information. If you go to the PEGS website, you can find these links. So we have the I2B2 web client. So you do need to request an ID. You don't need any DU or anything. You just go to the I2B2, PEGS I2B2 web page, you request an ID, and you can explore with the real data. It provides summary data. So if you're familiar with the All of Us platform, where they show summary information, like how many people have this disease, a cross section, how many people have this disease and are female, and those kind of things. So you can do a lot of data exploration. In fact, we encourage our collaborators to use the I2B2 web client for cohort building, feasibility analysis, etc., before they submit a proposal, so that they know what can and cannot be done using the PEG study. So that is with real data. And the ex-post results and the correlation groups, those are results from two published studies from our group, and those are also real data. So, and those are interactive visual graphics that show you associations with disease and exposures, correlations between different exposures as well.
Thank you. You mentioned that there were other phenotypes that you also, can you give a hint of which other phenotypes? And are you going to do other challenges, or are you going to use these phenotypes in some other settings?
Yeah, so to the first question, we have lots of different phenotypes in the PEG study. We have over 100 different phenotypes across all the three surveys. These are self-reported phenotypes. So participants were asked questions: has a doctor ever diagnosed you with such and such condition? So you see a representation of some of them here. So we have high cholesterol, blood pressure, allergies, asthma, diabetes, bone loss, and we have several questions on cancer and cancer subtypes. And we have many more phenotypes. Like I said, over 100, roughly 120 phenotypes in PEGS. Pregnancy complications. So we have all of these different phenotypes. We had a really successful challenge. I think we were really happy with the challenge results and the various models that the challenge participants submitted, and all the work that they put in, and all of the support from the Sage Bionetworks in hosting this challenge as well and providing their infrastructure. So we would love to host another challenge if you will let us.
I will allow you.
Great, thank you so much. We look forward to working with you again. In terms of, we are a very active group as well, doing analysis on many different phenotypes. So I have done several analyses on diabetes. We've been looking at high cholesterol, as I mentioned. We did a GWAS, XWAS, and a methylation analysis with high cholesterol. The XWAS results page actually goes through 15 different phenotypes. So I don't want to take up too much time in pulling that up, but if you go to the PEGS XWAS results from the PEGS homepage, we have XWAS or exposome-wide association study, which is similar to a GWAS, but with exposures instead of genetics. We have results for 15 different phenotypes that we show on that page. And we also show results stratified by age, sex, and race, self-reported race. And you can, break down those results however you want. You can look at the correlation groups to see for a particular phenotype how the exposures are associated with each other. So we are actively doing work on various different phenotypes. We also have many different collaborations. As I mentioned, if you submit a request, we do a DUA and share the data. So we have several collaborators across many different institutes like Mayo, and various other universities, and even within NIH, who are, they have their phenotypes of interest, and they are also actively working on various studies.
Thank you.